Welcome, everybody. Thank you for coming today. We are incredibly excited to kick off this new AI forum. So, we hear there's something about AI going on out there. It seems like there's a lot of interest in it. And so, this forum is meant to be a place where we can build a community of people who are very interested in AI. It's a nice place to us to get distillation of all the latest and greatest literature out there. And with time, this will evolve. So this is the first session, and so I think, again, we look forward to your feedback, and we're going to go ahead and evolve this over time to make sure it's of use to the broadest audience possible. I'm Abel Kum, a faculty here at Northwestern's Feinberg School of Medicine. I direct the Institute for AI in Medicine. And we're really excited today because Yuan Lo, Dr. Lo, is going to be leading the series. He's directs the new Center for Collaborative AI in Healthcare. And along with a really great student team and faculty advisors are going to be getting us off on the right foot. So with that, Dr. Lo.
Thank you, Abel, for the great introduction. So, ladies and gentlemen, and esteemed colleagues, and passionate advocate about AI for healthcare innovation. Welcome to the Northwestern Medicine Healthcare AI Forum. It's a dynamic platform where we immerse ourselves in the forefront of AI developments in healthcare. Here, we not only present the latest in published research and technological innovation, but also nurture open discussions among our attendees. Our forum is an inclusive space, open to the talented professionals across Northwestern Medicine and the broader research community from the Northwestern University and also across the greater Chicago area. Our mission is clear: to cultivate a thriving ecosystem for healthcare AI, one that encourages collaboration and supports a network of devoted scholars and professionals. Together, we aim to foster research, innovation, and leadership while seamlessly translating AI advancements into the intricate world of biomedicine. Our forum will be run by an amazing student organizing committee, including, let me click the forward link, Jenny Ding, Tanu Perea, Mario Alberto Garcia, Peter Graffy, Rishman Jivan, Dylan Prasad, Noah Forrest, and Zhang. To ensure that our forum constantly delivers the most pertinent insights into AI's multifaceted applications in healthcare, we have curated an advisory board of exceptional individuals. And these esteemed faculty members are from diverse disciplines, including medicine, engineering, art, and science. Additionally, we are honored to have the participation of senior leaders from the health system. As Chief AI Officer in the medical school, I'm proud to serve as the chair of this esteemed advisory board. We invite you to join us on this exciting journey of shaping the future of healthcare through the transformative power of AI. Together, we'll foster a vibrant community of scholars, professionals, and leaders who are unwaveringly dedicated to driving innovation in this critical field. Once again, I extend my warmest welcome to all of you. Let us embark on this exciting journey of discovery, collaboration, and progress in the realm of healthcare AI. Thank you for being part of this Healthcare AI Forum. Now, without much further ado, I'll turn the stage to the first student presenter. We also have a few standing mics on both sides of the aisle. So if you have questions, please feel free to line up and then ask when each presentation has finished. Jenny.
Thank you for the introduction. I'm very flattered to give the first presentation on AlphaMissense. So AlphaMissense, it's this new publication from Google DeepMind that's released in September, published in Science. And this is the same group that came up with AlphaFold about 2 years ago that caught headlines. So without further ado, let's get into it. This is the overview of my presentation. We're going to first go into some background information about what are missense mutations and current approaches for predicting the effects of them. Then we're going to look at the model structure, including structural context prediction for proteins, as well as some language modeling applied on protein level. And then for results, we're going to focus on first pathogenicity prediction on clinical benchmarks. So if you can look at the right, out of the 71 million possible missense variants in the proteome, about 32% are likely pathogenic according to AlphaMissense. And then we're going to compare the results with some wet lab studies. And lastly, we're going to look at some aggregate level, gene level results on whether a gene will be predicted as cell essential or not. And lastly, we're going to look at the contributions. So first, background. Before we get into the fancy modeling stuff, what is missense mutation? It is a genetic variant that causes a different amino acid to be encoded at a particular position in the protein. And then currently, we only know about 2% of them out of the 4 million possible mutations. And those of them are labeled as benign or pathogenic, and those are stored in the database called ClinVar. And traditionally, how we assess the effects of missense mutations is we have these multiplex assays shown on the bottom. So we saturate a protein with a bunch of amino acid mutations and measure their effects on cell growth. However, this process is a very labor-intensive approach. That's why we're going to have more machine learning approaches to target this issue. So firstly, we have this more traditional supervised approach of training directly on human curative variant databases like ClinVar. However, this is highly biased because we rely on prior knowledge. And there are some improved models, including training with weak labels. And what these weak labels are, it's that for a variant that's frequently observed in a population, these will be labeled as benign, and vice versa for pathogenic. And then we also have more language model-based approaches. For example, unsupervised modeling of amino acid distribution at a specific position, conditioned on the surrounding sequence context. And then we also have something like AlphaFold, which is relying on protein structure. And AlphaMissense, in our case, relies on the same models combined. Let's take a look at the model structure. So for the model structure, it looks complicated in the beginning, but what it essentially does is it takes a reference sequence, and then it does pairwise representation of amino acids. From this, it's capable of modeling the 3D structure, which gives out a structure prediction similar to what AlphaFold is doing. And then for the protein language modeling part, we have this reference sequence here. And in the second row, we have sampled variants, up to 50 of them. And for these black boxes represents masks, like language models. So we do masking on all possible variant locations. And then like other language models, we do random sampling of multiple sequence alignments. And whatever the model predicts as whether a amino acid is likely to be at a specific position, that will be the pathogenicity score. So it is measuring how likely a specific amino acid is at a specific position. So then once we have the pathogenic score, we do some fine tuning. And this fine tuning is basically based on, to reduce the contribution of rare variants. So what we do is for variants with high allele frequency and no pathogenic effects, we assign them with higher weights. And for those that are rare and benign, we assign them with lower weights. And then let's look at some results. So the first result is evaluation on clinical benchmarks. So on the top left, we see AlphaMissense outperforms all other models on the classification of ClinVar variants. These are the 2% that are labeled as benign or pathogenic. AlphaMissense achieve a high AUC of 0.94. In the middle, we have the calibration curve, and this is mainly to show that for variants that are labeled as pathogenic by ClinVar, these tend to have higher AlphaMissense score, and vice versa for the benign ones. And on the right, we see that AlphaMissense is able to increase the accurate prediction of variant effects by 25% from the next best model, shown as Eve here. And on the bottom, we have a specific example of a amino acid, ACVRL1. This is an important protein deemed by American College of Medical Genetics in terms of clinical actionability and prioritization for future MAVE. These are wet lab studies. And we could see that there is quite a lot of overlap between ClinVar labels, blue and red here, the dots here, and then what the AlphaMissense is predicting. And if you look at the structure, it makes a lot of sense. For example, the transmembrane domain that have a low pathogenicity, these tend to be less rigid in structure, whereas that compares with the globular domain, where things are more structured and rigid, tend to be more evolutionarily conserved for binding with its ligand. And let's look at some results comparison with wet lab studies. So wet lab studies, these are the main wet lab studies on variant effects that I mentioned earlier. So we have this database called ProteinGen. It has 1.5 million variants from 72 proteins. And then we have this prediction results on this specific SHOC2 protein. So what this protein does is it binds with PP1C and MRAS here to form a complex that activate the RAS MAPK cancer pathway. And we could see the result from growth assay here. So on the left, we have all possible amino acid mutations, and on the bottom, we have the sequence position of a specific amino acid, and we see quite a lot of overlap between AlphaMissense and MAVE. And that overlap is not observed in another model shown on the bottom. And looking at these specific sites that have high pathogenicity highlighted here, we can see that these sites shown here and here are really important for interacting with its two ligands, MRAS and PP1C, which explains more why they need to be evolutionarily conserved. And lastly, let's look at some gene level aggregated results. So one goal of the study is to assess whether a gene will be essential for human survival. And how we measure those traditionally is this measure called LOEUF, which is the predicted number loss of function variants observed divided by expected number under neutral selection. And this number is highly dependent on sequence length. So this gives out the problem that for some of the shorter genes, we can't use this LOEUF approach to measure whether a gene will be essential for a cell or not. So, and then here we have the, on the top, we have the AlphaMissense results, which had the best performance for these shorter genes that LOEUF is unable to predict. And looking at one specific example on the bottom, we can see that for this spliceosome protein, there's 7 subunits. The shorter subunits are highlighted in purple here. And although all 7 subunits are deemed as important from wet lab experiments, we could see that LOEUF wasn't able to predict their pathogenicity. In this case, lower number means is higher pathogenicity. However, if you look at AlphaMissense results, it's capable of predicting these short sequences as well as long sequences. And that kind of wraps up the main results of this study. And let's look at the main contributions of this study. So with this publication of AlphaMissense, a lot of resources being released to the biomedical research community. Most importantly is the prediction for 71 million missense variants, as well as their labels, whether they are clinically pathogenic or likely benign. Along with it is an aggregate level, gene level pathogenicity, which takes the average of all variants in a gene. And not only so, AlphaMissense also releases 260 million possible amino acid mutations across 20,000 human proteins. And lastly, AlphaMissense also provides the prediction for not only the canonical proteins, but also the 60,000 isoforms. And this can help us to study a full range of single residue perturbations in proteins and help us to better understand wet lab studies. More importantly, this AlphaMissense can help clinicians with diagnosis of rare diseases. So this plot shows the association between rare diseases, between AlphaMissense prediction of variant pathogenicity and its association with UK biobank traits. We can see that as the pathogenicity increases, it tends to be more associated with biobank traits. And this can help us discover previously unknown genes as well as their underlying complex traits. With that kind of wraps up my presentation. And thank you. Any quick questions?
Now we have time for 1 or 2 questions. The audience could just speak up if you can project to the whole room.
Yes, I
guess I didn't get why the pairwise.
Yeah.
Like, what are the pairs and why are the pairs?
Yeah. So there is pairwise representation because we wanted to. Oh yeah. So the question is, why do we have pairwise representation and why it's important for this model structure? So the pairwise representation is among every pair of amino acid. So if you know something about proteins, they have 3D structures, and those rely on long-range dependencies. It's not only the shorter context, but something could be far apart from each other and that contributes to the structure. That's why we do a pairwise representation, which is used for the structure prediction.
Oh, amino acid pairs, not sequence pairs.
Yeah, not sequence pair, but amino acid pairs. Yeah.
Thank you.
Yeah, sure.
Okay, since we have no questions, and we can proceed to the next one. And thank you, Jenny.
Yeah, thank you.
All right. Everyone, my name is Charles, and today I'll be presenting on the transfer learning, on the paper that titles Transfer Learning Enables Predictions in Network Biology. Okay. First, just a little bit background about these two authors. Christina, she's currently at Gladstone at UCSF, and her expertise is in machine learning, gene regulatory networks, and cardiovascular diseases. And the senior corresponding author on this is Patrick Eleanor, and he is a director of the cardiovascular study at BERT. So two very established people, and they've published many highly esteemed papers. Just a little bit background about why this project is in the shape it is. So there are many challenges in network biology, especially in the study of rare diseases and conditions that affect clinically inaccessible tissues. And this issue makes it a problem that if it can be modeled by computational approaches, that'll be great. And recently, there has been advancements in single-cell transcriptomics, and this technology has proven to be very powerful. It gives us a very fine resolution to look at the cell types at an individual cell level, to look at their transcriptomes. And this has proven to be very useful to understand the dysregulated pathways, genes in each of these cell types. Further, there has been many successes in transfer learning in, I suppose, in fields that's not just limited to biology, but especially in fields such as natural language processing and CV. And such success shows the potential in how pre-trained models on large-scale data sets can be fine-tuned for specific tasks with limited data, which is exactly the issue we're facing right now in network biology for rare diseases. And lastly, and that's another thing that's very good about this kind of model, is that biology often requires fine-tuning because different tissue, different cell types, different genes behave differently in specific contexts. This context can be patients, this context can be different people, can be race, can be many other things. And because of that, a model that has these fine-tuning abilities is a great platform to kind of access this computational ability and to solve such a problem. And the exact research question this paper is trying to answer is that given that we know gene regulatory networks map the drivers or facilitators of pathogenesis and allow for molecule screening in drug development, how can we accurately map such networks with reasonable generalizability, especially in scenarios with limited data? And their answer is a model called Geneformer. And this is basically a model that has pre-training down a very large-scale transcriptomic data. There's close to 30 million single cells from a variety of public data sources, as well as a variety of diseases, a variety of tissues. And the key thing is that this model has context awareness. And this context, just as mentioned previously, is disease, is tissue specific, and it's transfer learning. So we can transfer this knowledge with specific fine-tuning to specific scenarios that I will discuss in detail later. And in this paper, they also discuss its application to disease modeling, which is very useful for drug screening. The broad architecture and the overall workflow of this paper is that we started with single-cell transcriptomic data, about 30 million cells. And then instead of using the more traditional way of looking at the count data of these single-cell transcriptomic data, they actually just rank them by their expression within each individual cell. And I think this is a very clever way of doing this because in my personal experience and many of my colleagues, single cell data suffers from the noises and technical artifacts that we often observe when we look at different studies, when we look at different tissues, different people, et cetera. So I think especially at the data scale at 30 million cells, there's more than 200 data sets in here. The ability to have a non-parametric way of representing the expression of genes is actually very clever. And this ranking way of encoding the data is, I think, a key thing here in training. And then specifically within the encoder unit, there is the self-attention, which allows the model to look at different parts of the genes, different subsets of the genes at different contexts, so disease, tissue. And then it's able to output a few things that I thought to be very, very useful. So it outputs first the contextual gene and cell embeddings, right? So we have about 25,000 protein coding genes. This model is able to shrink that number down to about, I think, 256. So it's a much denser representation of a previously sparse data. And then it can be used to understand the interactions between genes and cells, which is what gene regulatory networks are what they're about. And then they have these contextual attention weights, which indicates which subset of features are being focused for specific tasks. And this is the I guess what the application of contextual information kicks in, is that it's able to kind of zoom in on specific subsets of genes when looking at different diseases, and that's very helpful. And then lastly, the contextual predictions, which can mean different things in different tasks. And there are different tasks being evaluated in this paper, and I'm only going to go into one of them in detail. But in general, it started by talking about integration and annotation, which is a great way to show that the embeddings that's generated by this model is based on true biological variants instead of technical artifacts. And the results from their integration shows that the cells clustered by their embeddings are based on cell type. So that's good, because those are actually biological variants and not by other technical variations, such as batch, such as experiments, such as reagents. And then they talk about gene dosage sensitivity predictions. So the Geneformer model actually correctly predicted the high confidence genes to be dosage sensitive in a specific context of fetal cerebral cells, which is a subset of cells, with 96% concordance with the original study. So this is basically kind of like a false, positive control. So we already know from a previous paper that there are genes that shows this dependence on dosage, and this model is capable to recapture that. So this is good. And then it talks about chromatin dynamics predictions. So this is talking about accessibility of the chromatin. And again, it correctly distinguishes genes with bivalent chromatin structure versus unmethylated structure. So very promising. And lastly, it's able to perform a network predictions. So it's able to distinguish Notch1, which is one of the paper that they previously published. So they understand a lot about this gene. It's able to distinguish the downstream targets without the perturbation data, which is, I think, very important, because perturbation, perturb-seq, if you will, has been kind of a very hot field, because it's able to predict, it's able to kind of sequence the changes in cell types when there is a certain perturbation introduced. And lastly, the in silico gene network analysis, which I will go into a little bit in detail here. So here, they basically show that how the deletion or how the interruption of two genes are able to be recaptured by the model. So on the left, we have a few targets. So there's Notch1 that we previously talked about, and the other one is NKX2-5 targets. And basically, when we disturb or in silico delete GATA4, we're able to see that the targets that are most affected are the direct targets of GATA4, which is what we anticipate. And then there are other dependent, like other genes that are facilitated by this, also show some sort of disruption. So it's able to recapture that. And then when they do a in silico deletion of GATA4 and TBX5, they observe an even greater changes, which is again very promising. So I think this is very significant, because oftentimes when doing perturb-seq or when screening for potential drug targets, is usually important. Usually knocking out one gene is not enough. So the ability to predict 2 or even 3 genes in silico and to screen for those potential targets in a model is very promising in a pharmaceutical context. So the ability for this model to show some of that capability is very promising. And this has been, so this is briefly what this paper is about. And I think there's many things that this model has shown to be capable of. This particular is what I'm most excited about, because the ability to predict 2 or 3 genes, their combined effect on the network is very, very translational. So that's good. And yeah, this is the paper. Thank you. Question.
Sure, you've seen those papers by David Platform and research, recent papers where they show like a limited reliability of those models in terms of zero-shot tasks like removal, subtype clustering, stuff like this. I'm curious, like, what are your thoughts? If this is like a major drawback of those models, or those models that just now.
Right.
Question, because section, so we don't mind.
Okay, so if I understand your question correctly, you're talking about the limitation or the, I guess, the low like reproducibility, if you will, of these or reliability. Right. So it's talking about the lowish reliability of the results. Basically, when we predict these, make these predictions, how reliable are they, or like how at zero? Okay. That's a very good question, and frankly, I don't actually have an answer for that question. I think it's still an ongoing field, and it's definitely rapidly evolving. I think what's important is that this is like in my personal opinion, what's important here is that because like even if we look at the concordance or the cosine similarity, the changes are not like drastic, if you will. So that means that the model is able to capture these variants, but the exact magnitude of them are not maybe not be like the absolute value of those might not be actually meaningful. But and I think that's why in practice, when we try to replicate this study, especially with true biological experiments, is that if we do like a Western blot, then it's going to be like very clear. The very slight changes as the one presented here will be much more difficult to quantify. And but I think that's like a limitation, but with I'm confident that with more data coming, especially right now it's at 30 million cells, I'm sure with the speed of the current academia and industry, like the speed that they're pushing out single cell data, this can quickly boom to 50, 100, 200. So I'm sure like at some point this will be a challenge that can be addressed. Yeah, but that's a great question. Yes, and indeed it is a problem. Yeah. Okay.
I have a super naive question.
Yes, please.
I have a super naive question, which is like, how big are these? I'm not in this field at all. How big are these networks, and like, how complicated are they? Sort of how connected are they? And
right. So it's hard to say like exactly how complicated they are, but it's so there are. If we go back, wait, how do I go? Oh, yeah. Okay. So I think there's like 6 transformer encoder units, and then within the self-attention, I think is a fully connected layer, and then I think the layer normalization is another layer. So yeah. Oh, it's trying to. Oh, I see. Okay, I would say it's very complicated. So just so yeah. So we have 25,000 protein coding genes, right? And within each of those, there are, they could be coding for transcription factors, and could be coding for like actual proteins. It could be coding for, many other things. Maybe they're not doing anything at all. Like the possible combinations within those 25,000 protein coding genes is immense. And some of them we don't even have any experiment data for, because the ability to our current knowledge on regulatory networks is mostly based on things that are more or less close to each other, right? So they're what they call cis. And if there are trends, which means they are more farther away, they could still be regulating per se, but it's very, very, very difficult. So right now, our experimental understanding is already limited. And to have a model that's able to learn it, not only on the stuff we already know. We wanted to like make predictions on things that we don't know, right? It's going to be several folds of magnitude even more complicated. And I think, which is why I think this is the most promising solution to the problem. And while it's not perfect yet, I think it's a valuable stride in the right direction.
Yeah. Computer science language, we are looking at a combinatorically large size.
yeah, got it. Thank you.
Yes. Yes. Oh, yeah.
So I was wondering, I haven't put too much thought into this, but I thought their in silico experiment was kind of interesting, that they used cosine similarity as a metric of assessing how well the in silico
Yes.
experiment went. So I think this means that they're assuming that the directionality of the vector kind of represents cell types. And I was wondering if you had any thoughts on like pros and cons of using cosine similarity as a metric.
Right. So I think this is a good point. So it's basically trying to measure. So cosine similarity basically measures like how different like the before and after is. And because it's already encoded, so each gene is already encoded into a 256 element long embedding. It itself doesn't mean anything per se, because like with counts, we can just count it, right? There's one transcript, there's two transcript. That's easy to interpret. At this level, it's a lot harder. And because of that, I think they're just trying to use a measure of similarity to see, okay, if I introduce this change in silico, how are the genes behaving before and after? And because of that, I think there really isn't a better way of quantifying it other than to say, okay, how different they are. But in practice, when we are like validating some of these changes, like with experiments, then we'll be able to see that. Yeah.
I was thinking more like
Mm.
instead of the embedding space versus technology. That's the another.
That could, yeah, I mean, it's a good point. Yeah, I mean, that would be you could be a reviewer. Yes. Yeah, I mean, that's a good point. Yeah, and it's not the most straightforward way of quantifying the changes. So, but there are, you know, practical considerations when they choose this. But yeah.
Let's just do a quick poll around the audience. So raise your hand if you identify yourself's interest as primarily bio side.
Like in the lab, like wet lab, who's more like on the wet lab side and who's more on like the health delivery or health side? We're just kind of curious. This is like a sparse
There's
sample.
16 people online, so could you talk?
Yeah, sure.
So if you identify yourself as primarily biological focused, please raise your hand
Just curious.
or virtual hand on Zoom or type it in the chat. Yeah, raise your hand if you identify your primary interest as bio focused.
Okay.
And raise your hand if you identify yourself as clinically focused. Okay, so we got a majority of people attending at least in person from the clinical side. But next time we hope that we will also have more people from the bio side coming in so that we have a free mix.
Okay. That's our guess. And so what we're going to try to do is, like I said, we're going to adjust as we go. But there may be like domains, topics that we're going to try to like sort of lean towards our themes. Yeah, sure. So as
This is our first session, so as we try to get a sense for what the audience is like, my guess, just like seeing this play out, is it's definitely more on the clinical or health delivery side, or biomedical side, a little bit less on the bio side. But as we saw, I mean, obviously the bio side is super important. It actually has really important long-term implications, but it might be that there'll be themed sessions that are focusing, so that, because we oftentimes see that there's a divide. There's the wet lab people and there's people that are not wet lab people. I may just put that ways. And if that's the case, then maybe it'll be more themed going forward. But it's just reading the audience, that seems like that's the case. Okay, thanks.
All right. Well, yeah. If you like clinical, I got the thing for you.
Yeah.
So my name is Peter Graffy. I am a PhD student in biomedical informatics here at Northwestern. I'm going to be talking about large language models in medicine, in particular one called NYU-tron, which is developed out of NYU. This is a presentation that was partially adapted from something done by Kathy Gao-Howard. She is going to be speaking next, and so you get to hear a little bit from her. I'd like to start just by talking a little bit about the evolution of large language models. The first one was actually developed back in 1967. It's called ELIZA. And there have been quite a few that have been developed between now and then. But really within the last 5 years, we've seen, I would call it like a renaissance of AI, especially in the large language model department. In this tree, the thing that I'd like you to pay attention to is just kind of there are two arms, the decoder only and the encoder only arms, which you can kind of see the brain branches there on the right and left. The decoder only arm is one you're probably pretty familiar with, stuff like ChatGPT, you've used it a bunch. And then on the left side, you'll kind of see the encoder only, which is typically referred to as BERT models. And we're going to be talking a lot about BERT models, but I'm going to kind of break down the differences between both. So you have an idea of kind of what we're working with. So when we're talking about BERT models, the B stands for bidirectionality, and bidirectionality refers to how it likes to read text. It goes from both directions simultaneously, a little bit different than your typical NLP models, natural language processing models, which typically only work sequentially or can work sequentially. BERT models are usually fine-tuned on smaller data sets. We're talking millions of parameters. They're highly effective for very specific tasks, text classification, and they're often used where text-specific language is common, like in the healthcare setting. Versus like a decoder-only model, that's more like ChatGPT, where it's trained on vast amounts of data. We're talking billions of parameters. They can learn a wide range of language patterns, and they can generate mostly coherent text in the way that you want. And the thing that I want you to take away from this, for the most part, is that BERT is really good at interpreting context, and GPT or decoders are really good at generating context. And so when we're talking about data that gets used, and we're going to kind of shift our focus to BERT specifically, we're talking about notes in a healthcare record. Notes are a great form of data that we can get access to. It's unstructured data, a little bit different than the structured stuff. And notes, there are a wide variety of notes, some of which are listed here and some of which you can see. So on the right, you can see kind of like a fairly typical discharge summary. And there's a ton of data that can be pulled from this using large language models and natural language processing. One of the first examples that really used, like it was a big data example, is Gatortron. This came about in 2021. It was a massive partnership between the University of Florida and NVIDIA. It was $70 million, took a bunch of years. And you can see all the clinical notes and everything that went into it. 290 million clinical notes, 2.5 million patients. It took a bunch of years, trained from scratch, and included PubMed, Wikipedia, and a bunch of other data as they kind of got it going. This required a ton of computing power and is not typically capable for most sites. But one of the cool things about Gatortron, you can actually access it on your own. There's a kind of web browser version that you can go and access. I encourage you to check it out. It's pretty cool. But what we're talking about for this presentation is NYU-tron. This is something that just got published within the last year, and just a couple months ago, actually. Based out of NYU, it's a pretty interesting one that's a little bit different than Gatortron, and I'm going to explain kind of some of the main differences. So for NYU-tron, we're going to break down kind of the methods or how they went about doing it. It started with 10 years of inpatient notes, about 340,000 or 380,000 patients, 4.1 billion words. They fine-tuned it using a couple different data sets, and then they gave it a few tasks that they wanted it to go after. They pre-trained it on a 109 million parameter BERT model, which we had talked about previously, and they called it NYU-tron. And they trained that on their entire EHR system. They fine-tuned NYU-tron to work on specific tasks. There's 30-day readmission, length of stay, and there are a few others that we're going to talk about. And they validated it on their held out data. And then they compressed it finally into an accelerated model. They loaded it in a form that could interface with their EHR system. And that EHR system actually could send email alerts to physicians for certain things that it was able to predict. And so the prediction tasks that I was talking about, there are 5 of them in particular: mortality, comorbidity index, which is the Charleston Comorbidity Index, 30-day readmission, length of stay, and insurance denial. For the purposes of this presentation, because we only have about 10 minutes and the focus of the paper, mostly they were focused on 30-day readmission, and that's what we're going to talk about a lot here for this presentation. And so one of the main things that they wanted to do was compare their unstructured data against structured data. And structured features have been known to be very good at predicting certain things, like pretty good, not extremely good, but pretty good. And structured is things you're familiar with, lab values, things that can be extracted out of an EHR. They wanted to compare that to the unstructured data that they got out of the notes. And so as you can see here, pretty much across the board, for their predictive ability, the unstructured data was outperforming the structured data to the tune of 5% all the way up to 14% for all the different outcomes they're looking at. So for example, we're talking about in-hospital mortality, 7% improvement in prediction, readmission, 5.36. So overall, it outperformed the structured data, which is pretty good. In the middle, you can see the Charleston Comorbidity Index, a little confusion matrix, and it correlated pretty nicely. And so when we focus a little bit more on the 30-day readmission side, one of the things that they wanted to do was compare against the human element. So they took out 20 patients, they had physicians, 6 of them, read through the discharge summaries and predict whether or not those people would be readmitted within 30 days. They compared that to what NYU-Tron could do. And across the board, NYU-Tron beat the physicians in every possible way. So true positive rate, they beat them by about 30%, F1 score, which is a measure of accuracy, beat them about 15%. So they were outperforming the human component, which is pretty promising. So then they wanted to compare it to other large language models, including kind of Gatortron, which we had talked about previously. And so there, like we talked about, there are many different large language models. And the one that they used, they kind of progressively ramped up the amount of data that would be required in order to do some prediction. And so you can see kind of the progressive add of the BERT base, and then they added in Wikipedia text, and then they added in PubMed abstracts and full articles, and then they finally added in the University of Florida notes, so huge models. And you can see in that table in the bottom left, the size of the model by the very end is huge, right? So we're talking billions and billions worth of words, it's take years for you to process through all that stuff. It takes a very long time to be able to work through it. And what we're finding here actually is that NYU-Tron was able to outperform kind of that basic BERT model that they initially started with by an order of magnitude, with basically just to get the 75% threshold, which is kind of seen as like a base threshold for predictability. They were able to do it with 1 order of magnitude less fine tuning, which is very good. That requires less computational ability, less power in order to be able to work with that. And that's really promising. And then the last piece was when they were doing some local fine tuning, they found that local was a little bit better than the external fine tuning. It helped improve their computational availability as well. And so the last piece that we talked about was their deployment in the EHR system. It interfaced with the system, and they wanted to see how well it did. They did that for about 4 months in 2022. They got about 27,000 patients, read their discharge summaries with NYU-Tron, and which represents about 1% of what it was originally trained on, so a very small percentage. And it found that it had a predictive ability of about 78.7% during that prospective trial. Of the 100 readmissions that it successfully identified, 61% were unplanned, 50% would have been a CMS penalty, and 27% were preventable at the time of discharge. And so the last thing that I want to kind of bring up about NYU-Tron is its overall limitations. So when you're talking about large language models and natural language processing, you can only have a finite amount of input that you can really put into the model. And those are typically referred to as tokens. A token is best identified as a thing, right? A token could be a character, it could be a word, it could be a number. It's kind of however you want to define it. When we're talking about tokens here for NYU-Tron, they had a maximum limit of 512 tokens. 512 tokens, when we're looking at our discharge summary that we had before, is about that. So it's not the entire note, is what we kind of came to realize, was that if you're not including the entire note, you're just going to be missing out on some data. There are other studies that I'm not including here, but other studies have shown that the more tokens you include, typically the better the predictive ability will be. It's unclear from the paper why they kept it at 512. There's probably some maximum computational something that they didn't really specify, but that's something that should be noted here. So the final conclusions that came out of this was that a smaller encoder language model that used only millions of parameters, this BERT base that we're talking about, was really good. It was fairly nimble and pretty robust, considering that you're comparing it against kind of the potentially larger GPT models that might require billions of parameters, or like the University of Florida one that required billions. It still required a ton of computational power in order to be able to do this. It is not a normal amount, and one that would be really hard to come by for most groups. However, if you are struggling with your computational availability, doing local fine tuning typically is better, and they found that in this case, that doing local fine tuning is extremely strong. And for the availability of NYU-Tron, you can actually go check it out. They have a GitHub repository, and you can go see some of the work that they did, so you can access it kind of on your own. Not with the same interface as Gatortron, but still relatively similar, so you can see how things worked. And so that is my presentation. I'll take any questions.
Hello. So like a lot of the notes and stuff in the medical records are full of like bias, especially one of the evaluation tasks in this paper was insurance denial. And having written medical notes myself, I feel like there's a lot of, inequities embedded into the information in the health system. And there are a few papers that recently came out about how large language models will only perpetuate these inequities. So I guess my question is, do you have any thoughts about that and like potentially how to alleviate that?
Yeah, that's a great question. So there are a variety of different ways that you can go about kind of combating what is just kind of inherent bias that comes with models like this. I do know there are even some people at Northwestern that are kind of looking to expand the generalizability of these. And part of it comes down to that you can't just work in the one system, right? It's fine for you to be fine tuning within, let's say, the Northwestern system. But if you want to be improving that kind of generalizability and reducing bias, then you're going to have to be incorporating more than just one type of data that you're going to be accumulating. I know it's a simple answer, but that's one of the better ways to do it is you need to have kind of a wider variety that you're incorporating in. So that kind of, is that kind of get what you're talking about, or are you talking about something else? Are you talking about the way that notes are written, the specific way that notes are written, like the structure?
Like, yeah, like starts with age, gender, and then it goes on. And so I feel like that's not necessarily something that is like amenable by increasing the sample size, rather like it's just how medical practice has been.
Yeah.
And so, yeah, that's sort of also been the question I've been thinking about recently too.
That's a good question. Yeah, I don't know. I bet that Kathy probably has a really good answer for that. No? Well, I for one am not a physician, so I don't necessarily know, but my best guess would be, I think, the understanding what the true bias outcomes would be. Like if what you're talking about in the structure does display some sort of bias that's affecting the ability to make predictions, I think that's a problem. But if a language model like this is becoming, I guess, smarter in the process of understanding what the structure looks like, and it's able to make better predictions because it knows how to anticipate the structure, that's not necessarily a bad thing in my book, if that makes sense. So becoming kind of habituated in a system and understanding what it's like in order to make better predictions, I think would be okay. But we can talk about it more after as well.
When that's a super important topic, it sounds like that should be a theme topic for future discussion. But having said that, I think there is a lot of like inherent ways we practice medicine. Like we're all sort of trained to do it in a certain way. I think one of the advantages, or one of the things that this technology brings us potential to, is to actually sort of reinvent sort of like every aspect of it. Like even the notes themselves can be deconstructed, I can imagine, in ways that are much more nuanced than they currently are set up in these very, very structured form. Like you said, we have a convention. When you're trained in medical school, you say, okay, the first thing you want to say when you describe a patient is, age, gender, race. That's what you start your presentation with. But is that the right way to do it? And it's hard to know. So, but again, sounds like a good topic for another future theme session.
Enter the physician to help with stuff.
Yeah, my question was pretty related, actually. I was just wondering, with the way that they truncated the notes to just 512 tokens, do you know if they like randomly chose the window for where they started and stopped that truncation, or was it always from the beginning of the note? Because like with the predetermined structure of notes, does that mean they're always missing out on certain types of information? Always only sampling for.
Great question. I believe that they're always starting from the top, working their way down. So it was like the first 512 that they're working with, as opposed to a section in the middle.
Okay.
Really great talk, Peter. Wanted to ask, why do you think the NYU-tron outperforms Gatortron? Do you think it's a product of NYU-tron being a BERT model and Gatortron being, I believe it's GPT? Or do you think it's a product of more so like data set shift, that it's really more so able to learn what's happening at NYU versus what's happening at Florida?
Yeah, I think a lot of it actually came down to their fine tuning and their approach to that. So the fact that they use fewer parameters overall for NYU-tron, and they kind of very specifically focused it at kind of a few targets, made it so that it was really good. And that's kind of what BERT models do, right? Is they're very good at, they're like savants at like certain things that you train them for. I think that the way that they specifically trained NYU-tron was a little bit more specific than for Gator. Although if I remember correctly, I actually think Gator is a BERT, that's a BERT base. Yeah, it had so many more parameters that it had, and it didn't have specific indirect endpoints, kind of like NYU-tron did. So I don't know if that answers your question, but it's.
Last quick question. I have a quick comment and a question. Comment on the previous thread. Actually, clinically, it's like various specialties are notorious for having different formats for their notes. Surgical notes are completely different than internal medicine notes. So I think there's also an opportunity to take that same work and apply it specialty by specialty, or at least within certain categories of specialties, and see how things perform. The question is perhaps for Doctors Ko and Lowe, but is any work like this being done at Northwestern to train a like gigantic model on all the notes?
We're doing that.
I think there's definitely more opportunity. And I think we do have to resolve some practical arrangement and legal arrangement, but I think that's what we're more sort of goes down.
All right, great presentation, all presenters. So good morning, everyone. Happy Friday. My name is Kathy Gao-Howard. I'm a critical care physician and research fellow. Thank you so much to the organizers of today's event for inviting me to talk about some of the work that my group has been doing looking at distinct clinical states. All right, I have no commercial disclosures, unfortunately. So, as you guys know, pneumonia, which is infection of the lower respiratory tract, is one of the leading causes of infectious deaths. It causes millions and millions of deaths per year, and this is the focus of my research. Something that I think about a lot as an ICU physician is why someone who comes in, say you have a 50-year-old woman with pneumonia, she requires respiratory support with mechanical ventilation. Sometimes people quickly get better, improve, go home. But another patient who might look very similar goes into renal failure, requires dialysis, actually needs to be put on lung bypass with ECMO, then maybe improves a little bit, but gets recurrent ventilator-associated pneumonias before eventually dying. Why is it that two people who look very similar can have such different diverging paths? Fortunately, the ICU, and especially the electronic health record system that documents all the data, is a really, really data-rich environment. And over the last couple of years, we've seen a huge explosion in a vast array of machine learning papers using this data to ask a variety of questions. Unfortunately, many traditional approaches focus heavily on admission and discharge, so how a patient are when they hit the door, and then whether they go home or die. Unfortunately, this really compresses their whole ICU course into a black box. And so that doesn't really help me coming on day 7 of service to tell me what I can do to really adjust your trajectory. Therefore, our goal was to develop a method that really allows you to unfold an ICU course to understand the contribution of intercurrent events. We hypothesized that clustering at the level of each patient day would reveal distinct clinical states, bringing us something from this to something more like this. So my work leverages the script systems biology cohort, which includes patients who are in the ICU, they're intubated, and they undergo a bronchoalveolar lavage for suspected pneumonia. I focus on the electronic health record data of this patient cohort, but the BAL samples do code to our lab and undergo a variety of multi-omic analysis, including single-cell sequencing. Furthermore, we have a rich layer of clinical adjudication where a panel of 5 critical care physicians reviews every patient chart and says, this was an episode of pneumonia, this pneumonia was cured or not cured. Suffice to say that there's a lot of data cleaning and checks, so I work with a really fantastic team to help me clean this data and get it into modeling shape. We had developed an approach called CarpeDiem to consider the patient's state for every day. So imagine a range of blood pressure. Sometimes patients can have normal blood pressure, sometimes they can have low blood pressure, be hypotensive, or sometimes they can have such critically low blood pressure that they require medications to keep them alive. We call that shock. And a patient might have a range of different blood pressures over different days. With our CarpeDiem approach, we would cluster similar days, so all the shock days, hypotensive days, and normotensive days together. We do this across 44 different features that are inspired by the practice of daily multidisciplinary rounds. Ventilator parameters, lab values, mental status, vitals, support devices. It's a cohort of 585 patients and 12,495 patient ICU days. So here, I'll show you our clusters. Each row here is a parameter organized in terms of organ systems, and each column here is a patient ICU day. So there's 12,000 here. We organized our clusters. We found 14 distinct ones, and we ranked them in terms of most favorable to least favorable. And if you look at this, you can start to see clinical patterns emerge. Red being high, you can see that there's a cluster here where patients have relatively high blood pressure compared to here, and many of these patients require vasopressor support. Here is a group, patients with relatively awake mental status, contrarily they're not intubated, versus clusters 12, 13, 14, where patients are all intubated, and many of them require ECMO for lung bypass support. Another way of visualizing this is with UMAP. So each dot here is a distinct patient ICU day, and we've overlaid it with our predetermined clusters that I showed you before. If you look at features, you can see these different islands emerge. So these two are patient days where the patient's intubated. This corner here, we call our ECMO island. Here you can see that days where patients require continuous renal replacement therapy all cluster together near the bottom of each respective island. And a more continuous value, D-dimer, which is an inflammatory marker, you can see is highest here, which makes sense because that's our ECMO cohort. And with this now, you can build a story and evaluate a patient's trajectory through clinical states. So here we have a patient and their ICU days down here. This is a patient who came in with severe ARDS, actually had a lung abscess, but was able to eventually wean off the ventilator. And you can see they move through the different clinical states here. Vertical lines are days where BAL sampling was performed. So not only do we have our clinical microbiology data for each of these days, for many of these, we have single cell sequencing results. And with this CarpeDiem technique, this helps give context to my friends in the lab to interpret what is going on and when they see their single cell signatures. If we take all patient days around an episode of ventilator-associated pneumonia, and we split them into cases where the clinical adjudication committee felt they were cured, you can see that overall, all cured cases have an upward trajectory toward better clinical states, compared to cases that were not cured, where there's a clear downward trajectory. More importantly, we can go back. So say these patients are both in clinical state 7 this day. Why is it that this patient improves to clinical state 2 and this patient goes worse to clinical state 11? We can even go back a day or two before and see if there are any clinical predictors that correlate between these transitions to better or worse states. Furthermore, it allows us to pivot to a variety of predictive modeling tasks. One area of interest that I have is modeling pneumonia cure. So starting with the day of the BAL, taking clinical information from days 1, 2, and 3, using a variety, just initial explorations been with ensemble gradient boosting tree algorithms to predict cure on day 7. Another task we've been working on with 2 excellent students in our lab is predicting next day extubation. So using features from today to say, hey, do you think this patient's going to be extubated tomorrow or remain intubated? And our models initially perform pretty well. We're looking to work towards actively deploying this in the EHR now. So overall, I think there's a wide variety and great opportunity to leverage machine learning on electronic health record data to help us. So when we see a patient on downward trajectory, we'll learn ways that we can divert them and help improve their care. So thank you so much to the huge team that I work with. Thank you all so much for being here, and happy to take any questions.
So how much difference would it have made, or do you think, if kind of periods have been more variable, unless it's day 1, day 2, day 3, but maybe something like periods marked by episodes of certain kinds that, you know, could plausibly constitute markers of transitions of some kind, and whether that would have made a difference in kind of the way you're telling the story in turn, or the kinds of predictions you're making. So in other words, variable periods based on particular events as opposed to just daily changes.
Yeah, so I think that's a really good question. And I think definitely to some degree, you can slice the data in a variety of different ways. You can focus on different pneumonia episodes. You can focus specifically on ventilators in pneumonia or community-acquired pneumonia, or even any other events. I think you can do it in a variety of different ways, and we can certainly, that's something we can explore further in future work. For here, we wanted to lay a foundational groundwork for every single day so that every single cell sequence sample would have a clinical context for it. I think that's definitely a direction for future work. Thank you for the question.